it's just a tremendous pleasure to introduce to our audience in the room and the audience remotely, Dan Roden, who is presenting our seminar today. Dan is famous to the world, so there's not much in the way of introductions, but there's a few things I'd like to point out. Is that, he originally trained in Montreal, and I love the fact that in his Montreal training, he did a year of private practice before he decided academics was his thing. Backstory to that. Currently, it's worth noting that he serves as the director of the Institute for Experimental Therapeutics, John Oates, who's also a famous therapeutics person and a very famous history of Vanderbilt around clinical pharmacology and therapeutics. Assistant Vice Chancellor for Personalized Medicine, Senior Vice President for Personalized Medicine, as well as Professor of Biomedical Informatics, and has served as Chief of the Division of Cardiovascular Medicine also during recent years. So it's trained in medicine, cardiology, clinical pharmacology, and in 2006 was really selected to spearhead precision medicine genomic initiatives at Vanderbilt, which are nationally, internationally leading and bring together some unique strengths that others are try to emulate and are often jealous of at the way they've succeeded. So the other point I note is that Dan has almost 600 publications, which are not in any way, individually lightweight. This is a huge volume of academic productivity. He is also co-PI on Emerge, on All of Us, Precision Medicine Resource, and on Genomic Learning Health System, as well as continuing to work on 2 R01s and PI of a T32. So, Steve, you're not getting out of the T32 for years to come. I'm just telling you. Steve's going, you got to be kidding me. But he and his colleagues, because it's a real team sport at Vanderbilt, have advanced the field of precision medicine, genetics, genomics, clinical genetics, and the use of the EHR. And we're just looking forward to hearing an update from Dan. And it's a tremendous pleasure to have you here, Dan.
Thanks for making that as short as you possibly could. I've asked a number of people what it is you want to hear about, and I didn't get much of an answer. So I'm going to sort of give a bunch of little vignettes, and the talk is probably a little too long, so I'm going to talk fast. Let's see. This is a Mac keyboard, so something I'm not used to. So I have to start by saying that I spent a formative year here in about 40 years ago. And that arrow points to the lab where I worked on the 7th floor of the Black Building, chained to an electrophysiology rig by this man, Brian Hoffman, who was chair of the Department of Medicine, or Department of Pharmacology, sorry, for many, many years, and literally wrote the book on electrophysiology. It's called Electrophysiology of the Heart. I have 2 copies, and I'm not giving either of them away. The other formative part of my time there is every lunch I would eat my lunch this way, from the hot dog from the soubrette guy, and what I did, how I spent my summer was learning how to do cellular electrophysiology and doing some work that implicated a mechanism for an abnormal rhythm provoked by quinidine, occasionally called torsade de pointe. And I could give an entire couple of hours on that, and I'm not going to spend any time or very little time talking about that part of my life. So I get criticized a lot when we talk about personalized medicine, because people say, well, we have lots and lots of big clinical trials, and they tell us how to take care of patients, and what you're trying to do is take people who are exceptions. And I think that those 2 views are actually not incompatible with each other. I think that most people are average for most things, and we need to do, as physicians, we need to just treat them as best we can, develop, from whatever evidence you have. But everybody in this room is far, far from average for some trait or traits. The trick is to figure out who those people are, find them, and decide whether you need to treat them differently or not. So, and the other part of this story is that in order to find small subsets of people who are credibly different from average, who you want to treat differently from average, you have to start with a very large denominator. So in order to find those little subsets of people, the little people in blue that are different, you have to you have to start with a big denominator. So what are the opportunities? The opportunities are to find people who respond differently to drugs because their pharmacogenetics are strange, or people who have unrecognized Mendelian disease, so you need to treat them differently from average, or people who are on these therapeutic odysseys because they have something wrong with them and you have to find out what it is. Starting in the neonatal intensive care unit, that's a place that there's a lot of interest in, but there are many people who have therapeutic odysseys even in adulthood. And then once you have these very, very large data sets, then you can start to think about subsetting common diseases. So common diseases like cancer or heart rhythms or dementia or whatever you want. Finding therapies that target specific individuals. Is there a way to get rid of the this thing? Yeah, thank you. Yeah. I'm not a Mac guy, so what do I know? So not only personalized therapies, but now you see it, new drug targets for weird diseases. And sometimes those drug targets are for weird diseases to start with, and they become targets or effective targets for common disease. So I came to Vanderbilt many, many years ago to do clinical pharmacology, the science of trying to understand variability in response to drug therapy in people. And I got immersed in this idea that there's a genetic component to variability very, very early in pharmacogenetics. So this is my way of showing that in cartoon form. That's a DNA molecule in Vanderbilt colors. The other thing that Vanderbilt made a big, big investment in early on was in the electronic record and in biomedical informatics. So this is a picture that Andy will recognize, George will recognize, but very few others will recognize. This is a medical record room. Those are actual patient charts. And you're taking care of a patient, and it's 3 o'clock in the morning, and you want the patient's chart. You have to hope that the record room is open. You have to hope that somebody can find the record. It's not misfiled. And you have to hope that it's not locked up in some resident's office because the patient was discharged 12 hours ago. So all those things are things that nobody who uses an electronic record even thinks about. But those are the kinds of problems that we had, not to mention handwriting that's illegible or stuff like that. So in 1991, our leadership invested in biomedical informatics by hiring one guy. And that guy created a plan for the development of biomedical informatics at the institution. And now there are 150 faculty in the Department of Biomedical Informatics, half primary, half secondary. I'm a secondary, but half primary, only in biomedical informatics. That was Bill Stead, for those of you who recognize the name. Oh, this slide. Well, this slide should have been animated and it wasn't, I'm sorry. But if you do a PubMed search, so in 2006, we had a strategic planning retreat and we decided to invest in personalized medicine. You do a PubMed search on Vanderbilt and personalized medicine, and you see the growth of references over time. I'm sorry that it's obscured. And the very first one.
When it thinks the cursor's at the bottom, it pulls up. So pull your cursor up a little bit.
Oh, my cursor. Oh, there. Oh, that's gosh, this is Max. Who knew? So the very first of these publications that you would get by searching Vanderbilt and personalized medicine is the description of our biobank, which we now call BioView or BioVU. So we started collecting samples on February 22nd, 2007. This is the sea of samples that we were collecting from. And the current numbers are almost 4 million people in the electronic record. I tell people that anything that is in the de-identified image of the electronic record is available to researchers, and anything that's not, it's not available to. It's because that's the phenotyping platform. And we're now over 350,000 DNA samples. And this is some of the numbers. I'm sure they're a little bit out of date, but you get the idea that the implications for the institution have been very, very large in terms of many, many projects, publications, NIH grants. The biggest NIH grant in Vanderbilt history was to support the Data and Research Center of the All of Us initiative. I'll say a little bit more about that if I have time towards the end. But I think that the biggest implication has been developing new kinds of science, developing new faculty. Some people come to Vanderbilt to play in this playground, developing trainees who have an aptitude for personalized or precision medicine. Some of those trainees are now leaders in the field. I mean, I've been talking to people this afternoon about Josh Denny. So Josh Denny started as a fellow in biomedical informatics, and has been a colleague for many, many years. So those are the kinds of people that we've trained. So the initial idea, at least my initial idea, was to use the electronic health record for genetic discovery. So you find a trait, you look at the dense genomic information that you have. Of course, it took a long time to get that information. And then you find where in the genome are there signals that tell you that account for variability in that trait. And those could be loci, they could be genes, they can be specific variants. So we have a tool which we call the synthetic derivative record counter. And I know that there were kidney doctors who I was going to see, but they're all at a kidney meeting today. So none of them are here, but the example that I've chosen to give you is a kidney example. So this is what the front page of the record counter looks like. Anybody at Vanderbilt can access this just without IRB permission or anything, because the question is, are there enough patients or enough records in the biobank for me to do a credible research project? So the example is chronic renal failure, and there are 43,000 people in the electronic record who have the diagnosis of chronic renal failure by something called a PHIWAS code, which I'm not going to explain very much of. Notice that this number and this number are different. It's not that we don't know how to add. There's a real technical reason why that is, and has to do with de-identification, and I can talk more about that. The next thing a reasonable investigator would do would be to click this radio button to see whether we have a DNA sample on this person, because this is the whole EHR. So there's 17,000 people with chronic renal failure and a sample. And if I then cross that with people who have array data, so we have about 100,000 people who have data on this particular array, the cross is about 9,500 people. So there are 9,500 people who have dense array data, this common variance, and chronic renal failure, and you can do a study on that if you're a kidney doctor and interested. You interested? There you are. Okay. So in sometime I was trying to figure out the timeline for this, but somewhere around 2022 we first announced, and then in mid 2023 we actually got underway something called the Alliance for Genomic Discovery, a partnership among Vanderbilt, Illumina, and entities in the pharmaceutical industry. There are now 9 drug companies that are part of this, and the major product will be to sequence whole genome sequence 250,000 samples. So before the alliance was created, the entire data set, dense arrays and EHR, you could fit it on your laptop. Probably fit it on your phone if you wanted to. But now with the whole genome data, we skipped terabytes, we're into petabytes now. So we have a lot of storage needs, and all that means is that the entire resource lives in the cloud and doesn't live on a server anymore. So now if you do the cross, we're going to get 250,000 whole genomes. Currently, there are 153,000 available, and there are almost 9,000 people with chronic renal failure and whole genome sequence data. So you could do something with that. And if you click here where it says click on more charts, you can see that there's a lot of ancestral diversity, and this is the kind of thing that goes right into your NIH grants if you're still writing NIH grants. So this is a cartoon from the strategic plan of the National Human Genome Research Institute, NHGRI, that was published 2020 or so. So we're halfway through this, and what I thought I would do is talk a little bit about some basic research, and then talk about a little bit about some clinical implementation, and then how the two interact. But every project that I will tell you about, and I have just a whole bunch of little vignettes, has a little basic piece and a little bit of clinical implementation piece. So bear with me. These are all examples of the kinds of things that the biobank can do for us. So obviously, the first thing that we did was do the experiment that we wanted to do, and that is define a trait, and find genomic loci and specific variants. So one of the nicest studies that I think that has come out of this was actually a long time ago, Wei-Chi Wei, who's now associate professor at Biomedical Informatics, did this study where he asked the question, who gets an acute coronary syndrome while they're taking a statin? So you have to find people who are taking statins credibly, believably in the electronic record, and then find them, follow them over time and figure out who gets an acute coronary syndrome event and who doesn't. Does a genome-wide association study, gets a very clear signal. A small study, but a very, very clear signal. And that signal is right on top of a gene called LPA, which is everybody's new favorite target for reducing coronary risk in patients taking statins. And if you go to the American Heart Association, you can get your LP little a measured if you're willing to wait in the long, long line of people, all of whom want to get their LP little a measured. So I was in a meeting in the office of our chair of biomedical informatics in 2009, 2010, something like that. We were starting to get enough samples that we were thinking about what genotyping to do, and we were doing a little bit of genotyping. And somebody said, well, you know, this is the paradigm that we've been following, but how about if we turn the paradigm on its head and say, well, here's a genetic variant, we have a curated electronic health record. Can't we ask the question, with what human phenotypes is that specific variant associated? And of course, that became the idea of a phenome-wide association study. I was in the room when the word PHOS was invented. It's a word that people use in genome science now. I did not invent it. I'm pretty sure I know who did. Pretty sure it was Dan Masis, because Dan Masis invented all those words. He was our chair of biomedical informatics, an acronym machine. So this is the typical PHOS, which is to say it's the best result we've ever seen. And Josh Denny drove this. And so the idea, there's a variant in a gene called IRF4 that defines or drives people having red hair and blue eyes. And so he asked the question, with what human phenotypes is that variant associated? And there's a very, very strong signal, P 10 to the minus 25th or something like that, with actinic keratosis, a skin condition. And then a strong association with a variety of non-melanoma skin cancers. And we were pretty happy with that. We validated that in eMERGE, in the Electronic Medical Records and Genomics Network, and in a paper in Nature Biotechnology, pretty high impact, we thought. But then we went on to a higher impact journal. Higher impact? Higher impact journal. And this is a really lovely article by Carl Zimmer who explains this complicated concept pretty nicely, and PHOS has entered the lexicon. So here's I sometimes hesitate to give this example because it's a little complicated, but for this audience, I thought I might try. So this is a complex example of PHOS, not a single genetic variant, but a set of genetic variants. So Nancy Cox, who was recruited to Vanderbilt in 2015, a very distinguished geneticist, statistical geneticist, generated this crazy idea. And I call it virtual transcriptomics. So if you wanted to study the relationship between gene expression in the liver, say, and a human phenotype, you'd have to do a bunch of liver biopsies. And you're never going to get to 100,000 liver biopsies, that's for sure. So she had this idea that she would take GTEx, which is a resource that does RNA sequencing in multiple tissues in recently dead people, and whole genome sequence. So she has that. And she can then generate multi-SNP predictors of the genetic component of variable gene expression. Does that make sense? I think it makes sense. And then she can do a PHOS using this as the input function and ask, with what human phenotype does predicted variable gene expression associate? So I asked her, because I'm an idiot, what gene is she going to study? And she looks at me because she's a statistical geneticist, and her answer was, well, all of them, of course. So here is the example that sort of proves the case, and that is a gene called GRIC5. I'd never heard of GRIC5. But when she did this analysis, this is the result of the PHOS. And what was remarkable is that there are these huge collection of eye phenotypes. of all kinds. And I've looked at lots and lots of these lists. They're sort of like looking at the telephone book. You just sort of see lots and lots and lots of entries, and you have to wonder, is there any significance to any of these? But seeing a lot of eye phenotypes was something very, very different. So she trots off to somebody else in the genetics environment at Vanderbilt who does zebrafish, and they suppress GRIC5 expression in the zebrafish. And as Nancy says, you don't have to be even a statistical geneticist can see that there's, this one only has one eye, this one has a big eye and a little eye, so there's something wrong with the eyes. And it turns out to be a vascular story. Vascular development, early vascular development of the eye predisposes to all of those phenotypes that I showed you before. And so there's this iterative kind of discovery in the biobank, back to basic science, back to discovery in the biobank. And the paper is cited right there. I couldn't resist. This is an example of torsade de pointe, and we're studying torsade. And one of the frustrations had been figuring out who in the hospital has that. And so Josh actually set up a system where every time the word torsade appears in an electronic record, as soon as the save button is pushed, I get an email. And the email says, we found this in this chart. And then we can go off and talk to the patient and get a biosample and do whatever things we want to do. And I'd love to talk to you about what we do with that, but that's another story altogether. So this is another longer story. Starts with a long QT story, but it could start with anything. This is an 11-year-old who had a witnessed cardiac arrest on the first day at school. He was resuscitated by an AED shock external defibrillator. And that, for those youngsters in the audience, that's something that 10 years ago, nobody would have been able to say, or 20 years ago. I mean, this is something new and it saves lives. It's not a normal ECG. And there are many diseases or a couple of diseases that this could be. We just sequenced him in-house because we do genetic testing in-house now. And he had two variants of uncertain significance, VUSs, and a gene called CALM2, which encodes a protein called calmodulin 2. There's actually three genes that encode exactly the same protein, calmodulin. It's a short protein. I'll show you how long in a second. These are the variants that never been seen before. So they're variants of uncertain significance. You can't use them in clinical decision making because you don't know whether they're real or not. So the VUS problem is this. So Andrew Glaser, with whom I've been working for probably a decade now, almost a decade, Andrew created this graph from ClinVar data. ClinVar is a data set that NIH maintains, and you too can look up, your favorite variant and see what annotation belongs to it. And in 2017, 2018, these are the numbers. There were some VUSs, some pathogenic or likely pathogenic, some benign or likely benign, and then some conflicting. And in 2024, the numbers look like this. That's because we've been doing a lot more sequencing of patients. And so all you do is get VUSs. And the VUS problem is actually worse than you think. So these are data from the UK Biobank, from the All of Us resource, and from BioView, the first 35,000 samples in BioView. And the problem is that, as you can see, there are about half of the variants, no matter where you look, are not even in ClinVar. And of the ones that are in ClinVar, most of them are VUSs. And what's the other interesting thing, which I like to point out, is that take any gene, take SCN5A, which is my favorite gene or one of my favorite genes. In the UK Biobank, there are 1,200 or so variants. And in All of Us, which is a data set that's half the size, there are the same number of variants. That's because All of Us has a focus on, I'm not allowed to say the word, right? Focus on diversity, in ancestral diversity. So, you see the same thing with BioView. So we did a curation experiment where we looked at the variants that had been reported for SCN5A. And at the time we did this experiment, there were 1,400 variants reported, 190 pathogenic, 91 benign, and the rest of them are VUSs. But I work with a guy named Fritz Roth, was in Toronto, now is at Pittsburgh, chairman of some computer science department. I can never remember and he did a back of the envelope calculation. He said, take the number of people on the planet, take the size of the genome, both known quantities, and the de novo mutation from generation to generation. And you can estimate that there are, for every SNP that is not embryo lethal, there are 50 or so humans on the face of the planet that have that particular variant. Now, we haven't seen them all because we haven't sequenced Madagascar or Sri Lanka or Paraguay. So I think we will see them all. Currently, in all of us, we've seen out of the 9 billion possible SNPs, 3 billion residues in the genome, so those 9 billion possible SNPs. Out of those, we've seen 1 billion already. So the problem is not this one. It's not the 1,400 variants. The problem is the 40,000 variants that are going to be there in SCN5A. And people are going to find them and then ask, are they pathogenic or not? So this is what I call a general scheme to assay function of all possible variants in the target genome. So bear with me while I just tell you, this is the kind of experiment that we are doing and other people are doing for many other genes. You pick your genomic region, could be a gene, could be a piece of a gene, could be an intronic region if you wanted to do it that way. You make all the possible variants, and then you either transfect them into cells that have been engineered to accommodate a single construct at a time, or you do this all in iPS cells using cell editing. So you generate a large pool of cells, each one of which has one variant in it, and then you challenge the pool with an assay. And the challenge is something I'll explain to you in a second. You sequence before and after. These are barcoded, so you can actually figure out which variant does what in the assay, and then you display the data as a variant effect map. So I'm going to show you examples of all of that. So here is an example of an assay that assays cell surface expression. So here's a pool of cells that have been generated. Some of them have lots of things on the cell surface, and some of them have very few, and some of them have none. So you sort cells by cell surface expression using an antibody, and you sort them into bins: low expression, medium expression, high expression, very high expression. And then you just count the bins. You do next generation sequencing, count how many samples from each variant end up in which bin, and then you can decide. You have an assay for cell surface expression, low to high. Here's another assay which asks the question: you grow these things under conditions favoring survival of a loss of function variant. So you might do it under a drug challenge, or do it under environmental conditions that where loss of function variants survive, gain of function variants don't. You grow them for a day or 2 or 3 or 4 or 28, and then look at survival, look at cell abundance by variant before and after, and then you generate a map. So this is a Calmodulin 2 map that Fritz actually had generated a number of years ago. And we used that map, looked at the 2 variants that we have in this kid, and both of them are pathogenic. And so we were able to move designation from VUS to pathogenic based on this, screen the rest of the family, and be assured that he had a calmodulinopathy, a disease, and that was his disease. And we've now published that, and we're doing this for major Mendelian heart diseases, heart genes. So here's a recent publication from one of my postdoctoral fellows, I shouldn't say postdoctoral fellows, MD Ph.D. student, who's now a resident in anesthesiology in Michigan. So she studied KCNE1, and this is what ClinVar annotates for KCNE1. The map is organized that this is the protein position, this is the amino acid position, and these are the protein variants that you could have. You have to look carefully. I probably should have pointed this out on the previous one, but every row is a different amino acid, starting with stop codons up here and ending with these guys down here. And so this is what we have now. So this is the map that Aisha Muhammad published last year. And she has 2 maps, a cell surface expression map and a functional map. This is the functional map. So we are very interested in the extent to which new AI-based predictors of structure, including AlphaFold, help with this kind of interpretation. And we're also very interested in understanding what the associated phenotypes are in the big biobanks like BioView or All of Us or the UK Biobank are going to help with that. So that's where we are with the variant of uncertain significance problem. Everybody likes to refer to pharmacogenetics as the low-hanging fruit. You know, the variants are common. They don't do anything to human physiology in general until you're exposed to a drug, and then you may get a terrible drug reaction or not. I think that they are, in that sense, they're low-hanging fruit. But boy, implementing a system like this to actually deliver on the promise of pharmacogenomics is a lot more complicated than I think I or anybody else gave it credit for. And it involves a lot of science and a lot of bioinformatics and a lot of engineering the health record. So our program, which we call PREDICT, started with clopidogrel and CYP2C19 variants, and now is up to greater than 25 drug gene pairs. It's an entity that's largely funded by the institution. This is what the report looks like. There's no identifying medical information on here, but I can tell you this is mine. And no, there's nothing very exciting here. But if I were a CYP2C19 poor metabolizer and somebody decided to give me clopidogrel, so CYP2C19 variants result in failure of bioactivation of clopidogrel to its active metabolite. So it doesn't, failure of efficacy is the adverse drug event. So if I were a poor metabolizer and I got clopidogrel prescribed, this is the pop-up that would appear. And the pop-up says, you sure you want to do this? Why don't you do something else? And physicians are always free to do whatever they want, at least in our system. So we've looked at whether physicians pay attention. And the things that make physicians pay attention are if the assay result is delivered quickly, if the patient is a homozygote, so they're more likely to have a severe phenotype than a heterozygote, the physicians pay more attention to that. And then things like age and the type of stent also influence their decision whether to change therapies based on the advice that they get. But the trouble is the biggest variable in determining whether a doctor pays attention to the advice that they're given is who the doctor is. So there are some doctors who look at it and say, okay, I'll change. And there are others who are complete naysayers and who will never change. And hopefully we'll be able to change that, but that hasn't changed yet. So I've talked a little bit about health records and genomics, but there are lots and lots of other things that determine individualized health, and some of them are listed here. Other omics, there's a lot of interest in that. There's interest in trying to figure out whether complicated medical regimens that people get are things that they follow or not. And often they're things that people can't follow because they don't understand how to take medicines. And there are other regimens that we as physicians prescribe to people, and they cannot follow that. I take medicines once a day, and sometimes I forget. And I refuse to believe that there are people who take medicines 3 times a day and other medicines twice a day and other medicines on alternate Thursdays that can be adherent to those kinds of regimens. So that's not the patient's fault, that's our fault. There are other things that determine individualized health, like how far away from a grocery store you live, what your socioeconomic status is. We're interested in using lots of interesting new data streams like images or step counts in terms of understanding people's individualized health. And I have to put in these iPS cells they're cardiomyocytes derived from iPS cells they're beating. And I have a whole story about that, which I'm not going to bore you with, but I had to put the picture in anyway. So the challenge is how to integrate all that. And so that's the challenge. Geographers like to do this. So I stole this from the geography literature. And that's one of the things that the All of Us Data and Research Center does. So our idea is to capture all the data in this resource, clean it, secure it, and make it available to the research community. So the current numbers approximately are that We're headed for 1 million participants. There are 850,000 or so who've signed a consent form. And not all of them have everything. So not all of them have supplied a biosample yet. Not all of them have supplied, not all of them have or have supplied electronic health records yet, and the like. There's a big focus on people who are underrepresented in biomedical research. That's not just ancestral diversity, but it's the rural, elderly, people who don't have internet access, those kinds of people. This is ancestry here, about half European ancestry and half non-European ancestry. And one of the things that really distinguishes this program from many, many, many others is that as a routine, the genomic data returned to participants. We don't call them patients, we call them participants. And the things that are returned are what I call recreational genomics. Do you like cilantro or not? Are you lactose intolerant or not? Those kinds of things. Ancestry information. But then pharmacogenetic information, hereditary disease risk, and it turns out, that most people, if you look at a lot of genes, most people will have a variant in a pharmacogenetic gene. And we find that, and everybody else who has these multiplex programs finds that as well. And so to date, there are around 3,000 patients who have received information that they have a pathogenic variant for one of these diseases here. We do the ACMG 59. I know that the ACMG count is now up to 81 or something like that, but there's a technical reason that we're stuck at 59. And that includes important cardiovascular diseases, important cancer susceptibility diseases, and other diseases. So I want to say a word about polygenic risk scores because that's everybody's favorite whipping boy now. So this paper, I think, brought the polygenic risk score idea to people's attention. So what this group of investigators did was they mined the UK Biobank, which was very, very large at the time and still is, and asked the question, if I construct polygenic risk scores for common diseases, type 2 diabetes, atrial fibrillation, breast cancer, coronary disease, can identify groups of people who are at the extremes of those distributions and quantify their risk. And the title sort of says it all. So they starts with a GWAS, and you generate a polygenic risk score. And the idea of a polygenic risk score is that people who are at high risk down here have a high burden of risk alleles. People who are at low risk have a very, very low burden of risk alleles and a high risk of benign alleles or less effective alleles. So, and this is an interesting paper because I think it expresses the idea in pictures really nicely, and it's from the cancer literature from 2010. So this idea has been around for a while, but now with big data sets, you can actually generate the polygenic risk scores, validate them, and think about executing them. So this is what the distributions look like. For these 3 common diseases, the risk goes up in the very, very highest centile. So type 2 diabetes, coronary disease. In coronary disease, which is where their focus was, you can see that if you want to find a population that's 5-fold increased risk of coronary disease, it's the top 0.5% of the distribution of polygenic risk scores. This is prevalent coronary disease. Then other groups have looked at incident coronary disease. That is what happens over time as people develop it. And this is an interesting and complicated slide. Let me actually, yeah. So if you just look at risk and ask the question, if you smoke, if you know about somebody's smoking status, diabetes status, hypertension status, family history status, you can generate a clinical risk predictor. And this is the performance of the clinical risk predictor with a C-index, C statistic around 0.68. Okay, not great, but not bad. Better than 0.5, which is a coin flip. You add in the polygenic risk score, and it goes up to 0.7. So it's a little improvement. It's not zero improvement, but it's not a huge improvement. And the other thing about the polygenic risk scores, which I think is important to think about, is that you can calculate a polygenic risk score in a fetus. I was going to say a neonate in a fetus. But you're not going to tell the fetus that their polygenic risk score says that they're at 6-fold increased risk of prostate cancer or Alzheimer's disease. You're going to do that later in life. you don't want, this is coronary disease, and you don't want to wait until they're 75 because then the difference between the low risk and the high risk group has already become apparent. And I think this is a way of measuring how effective the polygenic risk score is in prediction. What's the difference between this curve and this curve? So somewhere in here, somewhere in the 30s or 40s, people need to start to think about whether they're very, very high risk. And that may be where they start to mitigate that risk, if they ever mitigate that risk. So that's one part of the polygenic risk score story. And we're implementing polygenic risk scores in Emerge right now. What we're doing is we're generating something called a genomically informed risk assessment, or a GIRA. The idea is to generate polygenic risk scores, deliver them to families, deliver them to patients using not just genetics, but clinical risk like smoking or a family history of breast cancer, and then find people who are at high risk and tell them and see what happens. So it's not a randomized trial. There's a lot of reasons why that didn't happen, and I don't want to talk about those. I have a couple of other stories, which I think I have time to tell you. So this is, again, when I'm not being obsessed by personalized medicine broadly, I'm interested in the long QT syndrome. So there's a rare form of the long QT syndrome, so-called type 5 long QT syndrome. We were part of a consortium that looked at patients with type 5 long QT syndrome, 26 sites around the world, and we found 89 probands. So that's really rare. The commonest variant is D76N, and we had 9, we now have 10 probands who were apparently unrelated at Vanderbilt. We thought that was pretty weird because to have 9 or 10 worldwide for a really, really rare disease is odd. So we thought, well, are they related? So what we did was a study that used shared chromosomal segments and a technique called identity by descent. So the idea is, here is a person who has a mutation, the founder mutation, the de novo mutation. They mate, they generate offspring, and those offspring generate further offspring. And every time there's a mating, there's recombination events. So the chromosomal segment on which the variant sits becomes smaller and smaller as people are more and more distantly related. So you can see here are 3 people who have the variant, and their shared chromosomal segment is small. So the larger the shared chromosomal segment, the closer people are related to each other. So Piper Bilo, who's an expert in this, and Megan Lancaster, who is a fellow now, faculty member in my lab, looked at these probands, and basically they are all descended from a common ancestor that came to Tennessee or Kentucky probably 150 or 200 years ago. They're all 8th or 9th degree relatives, which is like the 2nd and 3rd cousins. I'm surprised they didn't know about each other, but they didn't. So that's interesting. But what was more interesting was that Megan then goes into BioView or Biobank and asks, is there anybody else with that shared chromosomal segment? You can define it by a series of shared, makes it sound small, but it's millions of base pairs that they're sharing. So there are hundreds or at least dozens of SNPs that describe that variant. So you can use the common variant backbone. And this is a map that Piper generated. At the time, she looked at 70,000 European participants in BioView, and there are 144,000 pairs of people. These are pairs who were related at the 6th degree. There are 3,400 who are 1st degree relatives. So parent, child, or siblings. So Megan asked the question: Are there what about those other people in BioView? And this is a distribution of normal QT intervals in our biobank. So we generated this more than 10 years ago, Josh and Andrea Ramirez and I, 30,000 people who have no heart disease, no drug exposure. So pretty clean set. And this is the distribution and the upper limits of normal as we and others define it. And we found 22 carriers in BioView, all of whom have D76N. And I don't know why there were more females than males, but that's the way it was. And some of them have normal QT intervals. and some of them have really long QT intervals. So the question was, why is that? And Megan then did an analysis looking at polygenic risk score for QT, and the people who have the longest QT interval have the highest polygenic risk score. So this is one of many, many, many examples now where high polygenic risk score in the presence of a rare variant confers a greater phenotype than just the rare variant or just the polygenic risk score alone. So I have one more story, which I think is interesting. This is a 29-year-old man who I saw 5 years ago in my clinic. So his family history is that he has an MYH7 mutation. That's the disease gene, one of the major disease genes for hypertrophic cardiomyopathy. The variant is known in his family. He has a cousin who is the proband, who has a hugely, massively dilated septum, has a lot of fibrosis, has had a myectomy for his symptoms, has heart block, has an implanted defibrillator. His mother has hypertrophic cardiomyopathy. She doesn't want a defibrillator. He has a brother with hypertrophic cardiomyopathy who does have a defibrillator, etc. He comes to my clinic with an outside echocardiogram. The echocardiogram is completely normal, with an absolutely normal septum and a normal ECG. And it's, 20, 21, 20, 20, something like that. And I said, well, in the year 2020 or whatever year it was, I said, you don't have hypertrophic cardiomyopathy, but we can test you anyway because maybe your kids will be interested or whatever. And then 4 weeks later, I'm at my clinic and I noticed his name is on my list. And I said to my nurse, I said, that must be a Vanderbilt scheduling error. I'm sure that doesn't happen at Columbia, but at Vanderbilt, we get people scheduling and failing to cancel appointments all the time. So she said, no, he's been called twice because he really needs to see you. And I said, oh, fine. And the reason he wanted to see me was, anybody want to guess? He'd been hospitalized twice in the 4 weeks between the time I saw him first and the time I saw him second for atrial fibrillation. Now, atrial fibrillation in a 29-year-old is pretty unusual, I will tell you. So he's mutation positive, has no ventricular phenotype, clearly has a phenotype. His MRI is normal. He's been followed. He looks like somebody with rare paroxysmal atrial fibrillation, and he has no hypertrophic cardiomyopathy phenotype. My colleague Ben Shoemaker did a study looking at almost 1,300 people with early onset atrial fibrillation. And we did whole genome sequencing thanks to the TopMed resource at NHLBI. And 10% of those people, 10 to 11% of those people have a rare variant in either a cardiomyopathy disease gene or a channelopathy disease gene. And the genes that are important are Titin, which was known. MYH7 is the second commonest signal. MYH6, which is a supposedly atrial specific myosin. LMNA, KCNQ1 is a channelopathy gene, and then many, many others. And the younger you are in general, the higher the yield until about age 60. I think that there are 89-year-olds who have a genetic component of their atrial fibrillation, but there are many other things that happen to 89-year-olds. So we now have a best practice alert in our electronic health record that pops up and it says something like this. It doesn't say something like this. It says this: your patient is young. They don't appear to have risk factors. Think about referring them to the genetic atrial fibrillation precision medicine clinic. And in the precision medicine clinic, they get deep phenotyping, MRIs, exercise tests, Holters, genetic testing. Part of that is clinical, and part of that is paid for by Ben's R01 that is looking at the question of, how important is any of this? Do we change therapy? Do we find people who have ventricular involvement? Do we find family members who need to be treated? Those are the kinds of things that we're interested in. So I think I've given you sort of a whirlwind of what we do in the personalized medicine space, how I think the field is going to evolve. I grew up at McGill, grew up in Montreal. I went to McGill. Sir William Osler, icon in clinical medicine, started his academic career at McGill and went to Penn briefly, then Hopkins, where he founded the Department of Medicine. He's famous for that. And then ended his career at Oxford, where he was the Regius Professor of Medicine. Anybody know what the Regius Professor really is? You would know. So it's a professorship endowed by the royal family. That's what the Regis, and that's the dean at Oxford has the title of Regis Professor. So he ended his career at Oxford, but his papers are all at McGill, not Hopkins, not Penn, and not Oxford. And I almost always close this kind of talk with this picture. So this is an interventional cardiologist, John McPherson, who actually directs our internal medicine residency program now, and a patient of his. And this was taken in 2010 when we had just started our pharmacogenetics prediction program. And we were really interested in finding people who were homozygotes for loss of function variants in CYP2C19s. And this is the first patient. And so John knows a lot about her, knows about her medications, knows about her life wishes. And now he knows a little bit more about her, that the clopidogrel is probably not the drug for her, despite the fact that she got a stent. So somebody said, Murdoch said at the beginning, it's a team sport, and these are the teams. And I could go through them, in gruesome detail, but I think the people to highlight are, the other rule, by the way, is that once you leave Vanderbilt, your picture disappears from this thing. So there are many, many people who have contributed to this effort over the years. Josh Denny, Dan Masis, other names that I've mentioned. They're not on here because they've gone away, but one of my partners in crime is Paul Harris, who's here and whom many of you may know. And Paul is actually the director of the Data and Research Center in all of us, and also the inventor of REDCap, which I bet you everybody in this room has used. And these two guys are my co-PIs for this genomic learning health system that we've just started at NHGRI. The idea is to figure out how to find people who have a genetic disease who should be evaluated and who aren't currently being evaluated. And these are my two co-PIs for eMERGE right now. So thank you. And I have even time to take questions.
Really fabulous. I'm Maya Sabatero, one question. So it feels like you created a lot of decision tools to help clinicians really understand when the trigger for pharmacogenetics, for example, should jump up. But it ends up being personal characteristics of the clinicians as to whether or not they're going to be using it. So I hope
that we'll get by that eventually.
Talk about how did you have you educated the clinicians about it? Did you conduct any studies with them to figure out what they're thinking? And also importantly, have you had any studies with patients to get their?
Those are great questions, and the answer is probably not enough. What I can tell you is a lesson I learned when we started the pharmacogenetics program. I came from a pharmacogenetics background in clinical pharmacology, and the data are pretty strong for CYP2C19 at least, although many cardiologists think that they're marginal. Even the AHA has sort of come out in favor of some kind of genetic testing, finally. So when we implemented, we generated this whole infrastructure on how to implement and how we were going to do the testing and what have you. And the mistake that we made was we never talked to the cardiologist. At least we didn't talk to the cardiologist initially and ask them, where should we do this and how should we do this? We talked to a couple, but we didn't sort of didn't address them. So that's the lesson, is that if you're going to implement, you have to go to the end user and say, this is what we're thinking. What do you think? How would that fit in? Would you be willing to use it? So the most recent example of this kind of thing, which is, for those of you in the cancer space, is a hot topic, is DPYD testing for patients about to get 5-FU or capecitabine. So there's a whole story around that, but there are variants in DPYD that predispose people to pretty severe, possibly life-threatening toxicity. And I was at a meeting earlier this week where somebody said, in the Mass General Brigham system, they've now decided to implement that. Well, that's very interesting. They decided to implement it because there was a lawsuit in Oregon where a woman whose husband died of this kind of toxicity sued the health system and came away with millions. So that's one driver of the uptake of that. So I talked to our colon cancer guys and I said, we're thinking hard about DPYD. What do you think? And they said, not interested. And I said, well, I got lots of things that I can sort of set my health IT people to. And if you're not interested, we're not going to do that. Of course, 2 weeks later, I got a phone call saying, I have Oregon and health sciences and lawsuit. Maybe we're interested now. So we're still arguing with them. But the point is that you have to engage the end user. And we didn't learn that. We learned that the hard way. Engaging the physicians, I will say that the cardiologists at Vanderbilt, when we started, there were a couple who were interested and most of them sort of shrugged it off. But most of them are sort of understand that it's available. And some of them use it for sort of what they think are high risk people. And I'm not going to argue with them because they have a pretty good sense of what's high risk and not. Like certain kinds of stents are not high risk compared to others. But I think we probably should do more. The patient education part, I think you have to, when we have focus groups and we ask people, are you interested in having genetic testing done for diseases? There's a whole spectrum of people who say, well, yeah, I'm really interested. I want to know about or I'm not interested. I don't want to know. But then you say, well, what about genetic testing for drug outcomes? And the answer is, well, you guys should be doing that right now. I mean, that's a no-brainer. Everybody, people don't disagree with that. People disagree with disease testing often, unless there's a reason in the family. So those are the lessons that I think we've learned so far. Nope.
So one of the, some of the last patient is really, hit me because I'm in cardiogenetics. And we see all these patients who come in with these like very highly penetrant malignant family histories, and they have the genotype, they don't have the hypertrophy. And looks like you have the scale there in your BioView system to look at, for instance, what does the father have? Is there a protective allele? So these phenotype negative, genotype positive patients of all hypertrophic cardiomyopathy, and whether or not there are these targets that I know drug companies are looking for in that patient. And he's just left with residual AFib, but he doesn't have hypertrophy.
Yeah, and he's probably not going to get it. He's a little probably old enough that he's not going to get a 3.3 centimeter septum. That's a great question. As you and I discussed, we don't have a great way of going back. We don't have any way of going back to patients in BioView, but in the clinic, that's what we do. I mean, we try to track part of the clinical ascertainment in the atrial fibrillation precision medicine clinic is generating at least a 3-generation family tree. And one of the things I get to do once every second Wednesday is I spend an hour getting presented cases from the clinic, and they present the exercise test, the MRI, the family history, the patient's history, and then we stop and you have to guess whether they have a genetic variant or not. And the family history is actually what sort of gives it away if they're going to have a Titan variant or anything else. So I think that accumulating families that way, as opposed to through the biobank, because as I told you, we can't go back. So we could figure out who's related to who, but that's a complicated exercise.
Going back, you could just have cohorts. I mean, if you use if you have whole genome sequencing on all these people, right? Cohorts of hypertrophic, of genotype positive, not hypertrophic.
And I will tell you, I should have used that example. There are thousands of people with hypertrophic cardiomyopathy in that collection. I mean, it's an amazingly common disease. About half of them have a variant, half of them don't. Yeah, no, that's on the list of things to do, on the long list of things to do.
There's an interesting, go ahead. No, you're good.
I'm Josh Marlowe from the pediatric ICU. I was wondering, thank you for this talk, if you could, your high throughput functional assay, the arc between publishing that paper, getting the annotation for something like VP, getting a bunch of clinical labs to buy into the value of that functional assay, and then getting many clinical labs to weight that to come out go from VUS to LPs, because it's great to see that on the screen. And I wonder about like my own
downstream.
When it's going to make it there.
So I don't think it's going to be lab by lab. I think it'll probably be ClinGen that drives this. ClinGen has a bunch of working groups. There are working groups that are focused on diseases. There are working groups that are focused on specific genes. And then there's a working group that's focused on how to integrate this kind of data into variant calling, and I think that's and the trouble is that people want a black and white answer. Is this pathogenic or not? And the answer is there are things that are really bad that are pathogenic. There are things that are clearly benign. We can't find a phenotype at all. And there are some variants that are a little bit bad. And no matter how you look at them, they're not normal, but they're not really, really terrible. And what's interesting about those is that the penetrance of those is really small. The penetrance of the ones that are, really dead channels, for example, is higher. It's not 100%, but it's much higher. So the pathogenicity of a specific variant is one of the things that determines penetrance as well. So I think we have a ways to go, but I think it'll have to be a national consensus around what is pathogenic and not, and then that will feed back into things like ClinVar, and then every lab will decide what to call or not to call, but they'll be a central source. And there's something called the MAVE consortium, and there's MAVE-DB, which is the multiplex assays of variant effect. So there's a consortium that does this. The NHGRI has a huge consortium that's doing this work. We're funded by NHLBI, and then the database is free for anybody, but it's not a useful database yet, I don't think.
There's one question remotely, so we will not cut it off yet, because that's unkind and unfair. It's an interesting question. Is there any analysis or project that you would like to do with the biobank data, but that is not currently possible because of privacy restriction?
It's not so much privacy. I mean, so the way the biobank is organized is that the clinical data are de-identified. The patients consent, but they get nothing back because it's de-identified. We'd have to break the de-identification algorithm. So there are things that we would like to do, like recontact patients. You have a really interesting phenotype. We would like to go back and do more deep phenotyping. Or you have an interesting genotype. We'd like to go back and do more. We can't do any of that stuff in the biobank. So one of the things that's exciting about the precision medicine clinic is that those are real patients who have consented to do anything, to recontact anything on a smaller scale than what you guys are doing here with everybody. So, every resource has its pluses and minuses, I think. And it's really important not to get married to the resource, but to ask the question, what's my question? And then figure out what the right resource is to answer that question. I think.
Well, with that,
That was no answer. Then.
One last question.
One last.
Yes, so how we can handle these, the overlap, common individual between biobanks. For example, all of us emerge and bio, if there is a contribute to this, all of them, how we will handle this in analysis?
There's a paper whose the first author was a woman whose name starts with FLAU, and I can't remember the rest of it. And the senior author is Alex Vick, who's from our place, that specifically looked at how best to analyze all of us and the biobank data together, and whether the right way is to do analysis and then combine, or do a combined analysis first. And the answer is, it looks like the right thing to do is analysis here, and then a meta.
Yes, but if there's a common, for example, not in UK biobank, but in emerge here in the United States, emerge all of us BioV. So some individual contribute to all of them. So someone is to BioV and also to emerge.
Well, the genetics will solve that, I think.
Yeah, but we can't do just that.
Yeah, no, I understand that. So yeah, I'm not sure I want to think about that problem too hard. I mean, the question is, are there people who are in 3 or 4 different biobanks? How do you figure that out? And if you're the one who's working on that, figure out a duplication algorithm, whether it's phenotype based or genotype based, that'll capture most of that. The numbers are very small. There are very few people I know in BioView who are also in all of us. There are some, but few.
Well, with that, I want to thank Dan. It was a tremendous tour de force over a life cycle or part of a life cycle. And thank you very much, Dan, for coming.
Thank you.